Thank you, Sean, for such an emotional speech that full of memories. And so I'm introduce myself. I'm Holden Liang. I'm a fifth-year graduate student in Dr. Ting Wang's lab. So and today I'm going to introduce you Dr. William Pearson. Dr. Pearson is a professor emeritus in the Department of Biochemistry and Molecular Genetics in the University of Virginia School of Medicine, where he served as the vice chair. Dr. Pearson received his Ph.D. from the California Institute of Technology, working in the lab of James Bonner. He joined the faculty in the Department of Biochemistry at the University of Virginia, where in collaboration with David Lippman, he developed widely used tools for comparing biological sequences. These include FASTP and the FASTA programs that enabled efficient searches of sequence databases. Dr. Pearson worked with Gary Stormo to organize an annual computational biology workshop at the Aspen Center for Physics. For his influential work, Dr. Pearson was named an AAAS Fellow in 2008. And now let's welcome Dr. Pearson.
Well, Sean's a pretty hard act to follow, and I haven't given an a cappella talk since I was in college, probably. So this is going to be more traditional. But first of all, thank you very, very much. It's a great honor to be a participant in this symposium. I have known apparently Gary not as long as some people, but for a really long time. And we've collaborated on a bunch of things. The Aspen Center, Gary was a stalwart member of the faculty for the Cold Spring Harbor Computational Genomics course for over, more than 20 years for sure. So it's really a pleasure and, as I said, an honor to be participating in this. So the thing which I thought I would talk about is how we got started with so little, because today we have so much. And I was actually, I didn't make a slide of this, but I was really impressed when the discovery of archaeal, the fact that the archaea exist. It's an exciting paper to read because there's almost no data. They didn't actually know how to do sequencing. They were doing RNA sequencing on a chromatogram. And somehow by looking in the spots, they put it all together. And all of a sudden we had a new kingdom of life. Well, I certainly wasn't part of anything like that. But when Gary started and I started, there really weren't very many sequences. And so I think it's fun to think about what that was like. So I don't actually, let's see how I can make this work. Whoops. All right. Boy, it really. Okay. So this sort of talks about the fact that a lot of the stuff that we know about sequences, we learned from very, very few sequences. So I've got the DNA stuff on the left and the protein stuff on the right. And it is really quite remarkable to appreciate how many hypotheses that were correct were being made based on practically no data. And so we had a Prigno box, a Tata box, was based on 6 E. coli promoters in 1975. I'm not actually sure they were E. coli promoters or if they were T7 or something like that promoters. We didn't have Maxim Gilbert sequencing until 1977. By 1982, Gary and his colleagues were starting to talk about what a ribosome binding site looked like. There were 124 of them known. Oh, thank you. And only 80,000 nucleotides of sequence known. By 1982, there were 2,000 sequences. But then people came along and they were starting to talk about what E. coli promoters worked. And then in 1989, Gary had a very influential paper finding a method for finding patterns in cyclo-KMP binding sites. Okay, that's the DNA world. In the protein world, we had a few sequences in the early 1950s. I was sort of looking back at old Margaret Dayhoff papers. By 1964, we had 50 proteins. By 1969, we had 300 proteins. And I actually just this morning saw a paper from 1961 where people were doing multiple sequence alignments from about 75 proteins and actually finding interesting things about amino acid substitutions and stuff like that. So at this point, I'm going to say that the whole idea of looking at sequences was a very specialized discipline. Margaret Dayhoff was doing it, Russ Doolittle was doing it, the DNA people were doing some kinds of things like that now. But it was really interesting to those people, but the biologists were maybe not paying so much attention until this happened. Okay, so in 1982, people realized that the Src gene from a virus was actually related to cyclic AMP protein kinase. They only had 1,500 sequences at the time, but all of a sudden there was biology there. There was something that nobody could have imagined, that there were these viral genes that were related to eukaryotic genes that did things that we appreciated. Things became more interesting. Likewise, in the protein world, there was this concept of homology, but people really didn't understand what that meant. What they understood was that there was excess similarity, and maybe there was excess similarity because they were doing the same thing, so they had to have functional constraints, or maybe there was excess similarity because they had actually evolved from the same protein. So there were some early papers on this, and we got scoring matrices in the mid-1970s. But there was this debate, as I was pointing out, that, so this is from a paper in 1968, where how can you not like a paper that starts out with a quote from Lewis Carroll? Do cats eat bats? Do cats eat bats? Do bats eat cats? For you shouldn't answer either question. It doesn't much matter which way you put it. And the question was, is excess similarity because homology, or is it simply there? And we can't really tell the difference between homology and excess similarity. So how do we understand things? So the argument that's being made here is that we know what excess similarity is. We can see things that are much more similar than they should be, even in 1968, but we don't know why. And there are biologists who say that we should say that this excess similarity is because of common ancestry. But common ancestry is an evolutionary concept. It's very specific. It's really saying something about evolutionary time that goes back along, now we know billions of years. And so they were very uncomfortable with that. We're comfortable seeing what we can measure, which is excess similarity. We're uncomfortable saying that's because of common ancestry. But today, we understand that because we've looked at so many sequences, that when we see excess similarity, we understand it to be common ancestry. So, but as I was saying before, again, this is kind of specialized until people started learning biology from it. And so the Barker and Dayhoff paper has Src genes being related to the cyclic AMP protein kinase. And then there was a paper by Russ Doolittle and Lee Hood and others where they were showing that a different oncogene was derived from platelet-derived growth factor or was related to platelet-derived growth factor. In both of these cases, there were very few protein sequences, and the only way that you got to look at those protein sequences is if you were collaborating with Margaret Dayhoff or Russ Doolittle, because their databases were proprietary. So then things started to change. GenBank was a great example. Sequences were just out there. You could search them, but we didn't actually have very good tools for searching them. So Wilbur and Lippmann published the SearchN algorithm in 1983. That actually caused me to start working with David Lippmann, collaborating with him. So he and I wrote the FAST-P program, which was published in 1985. In 1983, there were no public protein databases, but as the tools became available and the pressure because of GenBank, and there was a database called GenPep, protein databases started to become available, and we could start doing searches on them. And because cDNA cloning was happening at the same time, it was very exciting. You clone your cDNA, you do a FAST-P search or a FASTA search, and all of a sudden you might have an idea of what it was actually doing. And of course, in 1990, BLAST came along. Now we're up to 16,000 sequences. And people really got to learn a lot of stuff. So one of the things which I think is worth remarking is that all of the methods that we use today for doing similarity search, whether it's BLAST or HAMMER or SciBLAST and so on and so forth, are all older than a genome. Okay? They are all older than the human genome. Some of them are a little bit, we had the E. coli genome and we were getting the Drosophila genome at around this time, but basically these methods, which are still extremely widely used, are sort of back when we had practically no data. So this is a log plot of the size of protein sequence databases. And what you can see is that there are literally 1,000 times as many sequences as there were when HAMMER and SciBLAST were developed. And yet those are the methods that we use. And so I'm going to try to do a little bit of science here. So two things. So first of all, I want to say the reason that this stuff works so well, which is just totally amazing, is because unrelated sequences look like random sequences. So here are some histograms or plots of similarity scores against unrelated. Basically, it's against a protein sequence database. It's with 5 different sets of sequences. It's very hard to see on this figure, but there's a dotted line here and there's a dotted line here, which shows you what the distribution of scores is in the database. And all of those sequences are unrelated sequences. And then there are a bunch of solid circles here and here that show you where the related sequences are. And these guys have way more similarity than these guys over here. So that's why we can say when we have statistically significant similarity, we can infer the fact that the sequences are homologous, that they share a common ancestor. And this is the sort of the fundamental theorem of sequence similarity searching, which is when you find something that's not random, you can infer that it's related. And the reason you can do that is because unrelated sequences really behave randomly. And so that's a very powerful observation. The next thing I need to do is, because I have to do this whenever I give a talk, is say that DNA sequences are great. Gary Stormo's made an extraordinarily productive career out of them. They're really important if you're regulating genes and things like that. But protein sequences are where the information is. So what this plot is showing you is basically what I did was I took 100 human enzymes and I searched them against a whole bunch of proteomes, both at the protein sequence level and the DNA sequence level. And what you can see here is that at the protein sequence level, that's all these symbols over here, that even 3 billion years out from human to E. coli, using a single sequence, we can detect homology about 40% of the time. On the other hand, if you're using DNA sequences, which are these blue, purple circles, we get to go 80% of the time. We can find 80% of the enzymes in mice using messenger RNA. And after that, basically, we can't find anything. So DNA sequences allow you to look back maybe 100, 200 million years. Protein sequences allow you to look back 3 billion years. But even though we can look back a long way, we do not find everything. And that's where Gary starts contributing again. So here are a whole bunch of related sequences that do not have scores that are any better than the unrelated sequences. So these are what you would call false negatives. They're things we cannot find with a single sequence. So what do? We need to have a more sophisticated way of weighting scores. So when you do a BLAST search, do a FASTA search, do a Smith-Waterman search with a single scoring matrix using something called a position-independent scoring matrix, something that, again, has been around for a very long time based on a remarkably small amount of data. But if you really want to do things sensitively, then you need to use a position-dependent or a position weight matrix, which was a method that was developed by Gary for DNA motifs and published in 1988, and further developed by Steve Henikoff here in the audience a little bit later for protein sequences. And then it made its sort of life as everybody's using it in the current version of BLAST, which is SciBLAST, which puts these things together and generates the position weight matrix and does the searches with it. So as I'll show you in the next slide, these methods are extraordinarily sensitive. They can detect homology at very low identity levels, but they also have some problems where they're kind of tricked. So that's where I'm going to sort of move towards ending this. So here's what happens when you do a search using something like SciBLAST or some other methods that hammers up here. Another method that a modification of SciBLAST that I developed is up here. Basically, if you just do a single sequence search, you're down here, and perhaps you're finding 10% of the homologs to be found. Whereas after you've done a few iterations and you develop your position weight matrix, then all of a sudden you're able to find, perhaps 95% of the sequences, the homologs that are in the database. The problem is that as you're making your method more and more sensitive, you're also generating a lot more false positives. And the reason that you're generating the false positives is because of something called alignment overextension. You're basically corrupting your position weight matrix. And there are some tricks that you can use to allow you to keep the false positives down. So there is hope. And this is another figure that sort of shows that. Basically, when you do a SciBLAST or you do a hammer, you start out with there's a homologous domain, but unfortunately the alignment doesn't just stop over the homologous domain. It can extend into something which is not homologous. And once that thing which is not homologous has been extended into, then the position weight matrix has been corrupted, and you start finding things which are always wrong. And unfortunately, it's really hard to see when that happens. So again, these methods were all developed when there weren't any sequences, or at least there were a lot fewer sequences than there are today. And one might ask the question, how come we can't do better? So this is one way of thinking about it. If you think about protein families as little galaxies or stars, in 1984, we didn't have very many. By BLAST and SciBLAST, we had a lot more. Today, we have 250 million protein sequences. So the obvious question is, why don't we find everything? And I'm going to leave that question as sort of an open one, because we still don't have a complete answer, unless, of course, I guess I didn't point it out to you, but AlphaFold was up there on the graph. Now that the databases are very, very large, we have problems because they are spectacularly redundant. But because they're so large, because the stars are now, the protein universe has been sampled so thoroughly, the question of sensitivity is a little bit less important. And getting the alignment exactly right is actually much more important. If you can get the alignment right, then you don't have these overextensions and you don't have these false positives. And of course, the other thing that we really care about is not who's homologous to who, but what are the actual functions of all these different proteins. So, and the other thing which is sort of not well appreciated is that we search databases where a choice has been made as to the sequence in the database, and oftentimes that's actually not the best choice. So here's a case where this sequence and these blue sequences were in fact the sequences in the database, but there was an isoform of this cow sequence which would have made things much better. Now I'm working with the UniProt group, and we're actually trying to correct that, so hopefully that will be less of a problem. So we have lots of sequences. We can still do better. We can do better by paying more attention to getting the alignments exactly right. But again, I want to thank the organizers and thank Gary he's been a really great supporter over the years. And these are the people that I work with.
Questions for Bill? Just raise your hand and a mic will come to you.
So while I've got this quiet and you're moving mics, I'm going to show you two more slides, which is where the FASTA format came from. Okay. In 1983, these were the database formats that were known. Okay, there was something called the EMBL format. There was something called the GenBank format or GenPept, and I was working, or David and Libin and I were working with Margaret Dayhoff, and they had this this other format, okay, which had a greater than sign, and it had a description of the protein, and then it had the sequence. And when we wrote FAST-P, those were the sequences that we were looking at. So the problem with this format is that these formats are horrible because they've got all these columns that matter and special labels and all sorts of stuff, which makes things really hard. This was a great format because it had a greater than sign and it had this little description thing, but it had this description line that basically everybody forgot about. So what they would do is they would give you a sequence that looked like that, and then FAST-P would basically throw away the first line because it was supposed to be the description. So my contribution to computational biology is that I took this format and I made it into this format.
Yeah, thank you very much for inventing the FAST-A format. Makes us work much easier. So when we grep stuff, we can just remove the header. And my question is there still application for like FAST-P or FAST-A in today's work?
So I don't think there's much use for FAST-P, but FAST-A does its statistics differently. It does empirical statistics as opposed to sort of the model-based statistics that the BLAST programs use. So if your sequences are appropriately weird, then FAST-A may give you more accurate statistical estimates. Okay, and the FAST-A also has a FAST-X and a couple other things where you do searches with proteins against DNA sequences. BLAST has the same thing called BLAST-X. I'm going to say FAST-X is better. The way things are, we're doing the alignments and stuff like that. So I'll just say another thing for those of you who are trying to annotate genomes. The best way to annotate genomes in a day when we have so many protein sequences is to just search protein sequences against your genome. FAST-X or BLAST-X will be happy to translate your genome for you. You do not have to look for open reading frames. Looking for open reading frames is actually counterproductive. And then having done that, you'll find matches to your proteins because all the proteins are, except for, I'm going to say some, like archaeal bacteriophage that live in volcanoes or something like that. Pretty much the protein sequence universe has been sampled. Anything else? Well, thank you very much.